Boom. All right. I am Nora Serbow. I'm the president of the Hardy Plant Society Mid-Atlantic Group, and we are so pleased to welcome everyone here today. The program represents a core principle of the Hardy Plant Society, and that's collaboration. And in this particular case, we're delighted to be partnering with the Haverford College Arboretum and the Henry Foundation for Botanic Research. And I'm especially pleased that we have Mike Aucott, who happens to also be an old friend of mine, but he also has Haverford connections as our speaker. In addition to this important collaboration with Haverford, we also partner with other public gardens and organizations throughout the Delaware Valley. On October 15th is the Perennial Plant Conference, which HPS jointly sponsors with the Scott Arboretum, Pennsylvania Hort Society, Chanticleer, and Longwood. On November 14th, we collaborate with the Scott Arboretum in a joint lecture, similar to this joint lecture, with Tony Aiello, the gentleman pictured in the middle, from Longwood Gardens. He will speak on the role of botanic gardens in tree conservation. On December 5th, we have our annual meeting, hopefully at the Jenkins Arboretum, with speaker Janet Novak on wildflowers of the Mid-Atlantic region. She's a member of Hardy Plant and 11 other plant-related societies, ranging from the Rock Garden Society to the Rhododendron Society. She also has photos that have appeared in Horticulture Magazine and in the Peterson's Field Guide to Ferns. Now we have it. We also organize a variety of what we affectionately call pop-ups, which include tours of public and private gardens throughout the region. This year we have pop-up visits to our grant recipients as well. I believe this month we are going to visit the Tyler Arboretum, which received one of our grants. The work being done to enhance, educate, and beautify the region is a satisfying aspect of our organization, and the visits are a treat for our members and our grant recipients. Each year, Hardy Plant Society awards grants throughout the region. Our grants have been distributed to schools, parks, arboretums, and public gardens up and down the East Coast and as far away to the west as Pittsburgh. The gardens also go to municipalities, and many, many more. The grant application process begins December 1st. So if you know of a worthy project, and visit our website to get the details. And for those who love to continue to learn and have enjoyed our winter webinar series, Tuesdays at 10 comes back in January. So keep your eyes on our website, hardyplant.org, to see what will be coming then. We have a wonderful array of events planned for 2022, including a seed exchange, which is for members only, and our March into Spring Symposium, and our hugely popular members only garden tour, which I understand will be in Montgomery County this year, with a reception at the end of June. So we have tours, both domestic and international, planned for 2022. If you're not a member, we encourage you to join in the fun. Just visit our website. And now I am so pleased to have this program with Haverford and Henry and to welcome Mike. And with that, I will stop my share and hand this.
Everyone, my name is Jenny Sobrowski, and I am the program coordinator for Haverford College Arboretum. If you aren't familiar with us, we are located in Haverford, and we are a 216-acre college campus. It's open and free to the public. We are happy to offer it back to visitors this year after a long closure due to COVID. And if you are interested in joining us in any future programming, please feel free to check out either our Facebook, Instagram, or website. We offer both virtual and in-person programs and plan to continue to do so throughout the rest of the season, hopefully continuing back with in-person programming in the spring semester. We are very excited to have Mike with us here today. Mike is a Haverford College graduate, class of 1968. So we always like welcoming alumni back. He is also part of the science committee for the American Chestnut Foundation and a member and the environmental commissioner of Hopewell Township. If throughout the presentation you have any technological issues at all, please feel free to email me and I will try to get you back into the room. We have not had the wait room enabled throughout the presentation, so if you need to leave and join back, that's completely fine. And we ask that you use the chat room for your questions. We're going to leave room at the very end of the program to go over anyone's questions that they may have. So without further ado, I'm going to turn things over to Mike. Thank you, everyone.
Thank you all. It's really a pleasure and a privilege to be able to talk about chestnuts again. I love to talk about chestnuts, and there's a lot to talk about. So let me get into this presentation. And so here's what I want to talk about, kind of an overview of this. It was a very high value tree, excellent producer of strong rot resistant timber, reliable nut producer, fast growing, widely adapted, nearly wiped out over a century ago. Early efforts to bring it back really did not succeed. There are a number of new efforts underway, and they're based on a better understanding of genetics than we had back in the day, and increasingly are incorporating new methods. Nevertheless, the effort to bring this tree back to its majestic presence that it once had will take decades, probably centuries. So we're in a long process here. We think of a big tree, we think of an oak. What's the mightiest, biggest tree around? But the oaks once had a big brother. And here's a wood engraving from the late 1800s, harvesting chestnuts in Fairmount Park. You might think that the artist exaggerated a little bit. Look at this little tiny people and the great big tree, but here's some actual photos. Down at the bottom here, you can probably see a person, and that tree is 10 feet in diameter, and this is how they grew when they were in the open. Here's another one. In the forest, they grew tall as well. Certainly a canopy species. Here's another picture, and another. This one is interesting because it shows the tree having sprouted from a stump. You can kind of see the remnants of the stump there, and this was one of the attributes of the chestnut that made it especially valuable as a timber tree. It would grow back very quickly from the roots. And in fact, the dominance of the chestnut, it probably made up at least a quarter of the forest in many areas. Probably had something to do with its ability to sprout from cut stumps, because as we know, the colonists clear cut vast areas. Like New Jersey was probably totally clear cut in the late 1700s, early 1800s. And those clear cut areas became good growing places for chestnut. It came back stronger than most other trees, but it was a very common tree, and certainly produced large logs. This is a workbench that belonged to my great grandfather, and this is chestnut, and you can see the small growth rings. This is a big piece of wood, and it was probably the dominant timber tree in Pennsylvania in 1911. You can see the percent of timber from these various counties that was chestnut. This is a lovely old book that I was lucky to get a chance to get a copy of. First published in 1915. This edition was 1928. And in it, the author says that it was formerly the most common tree in Pennsylvania. Here's a picture of what a hillside looked like when the trees were in bloom. This is probably Shenandoah Mountains in Virginia. And at this point, I want to take a little break and sing you all a song. I think that when we're trying to deal with the chestnut, understand it. trying to understand what another species is going through, and what would the chestnut be saying to us, calling from the past through photos like this, calling to us in the present. And what would it say if it was doing that? Well, here's a song that was written by the wonderful songwriter and singer Laurie Lewis. Laurie Lewis is based in California, but she visited the Appalachian Mountains and heard the story of the chestnut and wrote this song from the perspective of the tree. What might the tree be trying to tell us if it could? There are none still alive who remember all along the Appalachian chain how we once ruled the woodlands, the surrounding hills and plains. Shawnee, Cherokee, Cohan, and Creek, we answered to their needs, and we asked for nothing in return but to plant and to spread our seeds. When the plague it came among us from across the wide empty sea, weak and strong alike were fell, for it knew no spark of mercy. Some say we're gone forever, and there are so few who care. The oaks feed the few who still linger here, the squirrel, the jay, and the bear. But we bide our time beneath the earth, who once were giants on the land. Each year we send up our emerald shoots, and we will rise again. Each year we send up our emerald shoots, and we will rise again. So that's Laurie Lewis's song called American Chestnuts. If you don't know about her, I encourage you to check out her website. She's written some beautiful songs. I think that's one of them. So the blight came along. This is the next chapter in the story. It was a widely adapted dominant tree, and then the blight showed up. The blight was actually probably arrived in the late 1800s, but it was really identified in 1904. Its spores were spread by the wind, by birds, by insects. It advanced steadily, and as Laurie Lewis said, weak and strong alike were felled, for it knew no spark of mercy. By the 1940s and 50s, the blight had pretty much killed all the trees throughout the tree's range, and it left this kind of scene in its wake, ghost forests. The tree is not extinct, though. It still survives in the understory. There's millions of little sprouts. You can see them along the Appalachian Trail and other places. But they usually die as they succumb to the blight before they're about 20 or 15 feet tall. And the tree is no longer present in some areas where it was once plentiful. It's gradually fading out. This is what the blight looks like when it gets started. You can see the orange sporulation here, and you can see that the blight it's kind of sunken into the trunk. And what it does is it girdles the trunk and stops the flow of nutrients and moisture and so on, and everything above that dies. And doesn't take much for the blight to get started. The spores are everywhere, and any kind of nick or ding or a vole bite in the bark will allow the blight to get access. And if the tree has no resistance to it, this is what happens. And you see in the woods often chestnut sprouts, as I said, and here's one that had grown for a while and now it's dead. But you can see it's sprouting vigorously from the roots because the roots aren't killed by the fungus. And as long as they can get some solar and you know, they can survive, although they're gradually fading away. So what are some of the challenges in bringing this tree back? Well, there's a number of them, but probably the two biggest ones are that it has to be, you have to have a tree that can tolerate the blight, and it has to be able to reach the canopy, because if the chestnut does not bloom unless it's in full sun, if it doesn't bloom, it won't produce seed. It's not self-fertile, so you have to have at least two trees that are close enough to each other to pollinate. And that's a big challenge. These sprouts cannot do that. They just don't live long enough to get into the canopy. But back when the blight first started taking hold, the idea was to find trees that were related to the chestnut, close enough so that they could hybridize, and cross them. And one of those trees is the Chinese chestnut, the Castanea mollissima, and another is the Japanese chestnut, Castanea crenata. And these are beautiful trees. They're wonderful nut producers, but they grow like an apple tree. They're suited for an orchard environment, and they probably have been bred that way for thousands of years. And the trees we have access to in this country, at least, grow like apple trees, and consequently, they cannot reach the canopy in the North American forest, and they will not bloom in a forest environment. There's another related tree, and that's the European chestnut, Castanea sativa. It does have the tall growth habit of the American tree, but it's not very blight resistant. So there's a problem. None of these trees are ideal for crossing with the chestnut. Or what typically happens when you create a hybrid by crossing a mollissima or crenata with dentata, which is the scientific name of the American tree, you get a tree that, like the ones on Haverford campus, the wonderful Haverford campus that I was so privileged to spend some time on back in the day. We have a couple hybrid chestnuts on the campus, and they are crosses of dentata and mollissima, but they have a characteristic growth form of the of mollissima. Here is one of them, and here's the other one, and you can see it's got a branchy growth habit. It's not going to, and these trees probably aren't going to get too much taller than this. They'll probably max out around 50 feet or so. And they can't compete with a guy like this one in the back. Of course, this is not a native spruce probably, but the forest canopy is going to be up at this level in North America, and these trees aren't going to be able to get there. Hybridization efforts had begun in the 60s or actually began in the 20s, but were pretty much phased out by the 60s. And it was mostly because arguably there was not a full understanding of genetics at the time. Gregor Mendel, who many of us studied and had to learn about Mendelian genetics, published his papers in the 1860s, but they were largely ignored. He was kind of at the periphery of accepted scientific circles and wasn't paid much attention to. But his work was rediscovered in the 1900s, but didn't really come into wide usage until the 30s or later. One of the but I just wanted to give you a little quick run through about genetics. We'll use an example of eye color. It's unusual in that it seems to be controlled by one gene primarily, and there are two alleles. They separate, they recombine on reproduction. The gene for brown eyes will give a big B, and for blue eyes a little b. And the big B tends to dominate. If a person has one of these two genes, then it's a big B, they'll have brown eyes. And you can consider that if both alleles are the same, that we call it a homozygous genotype. If they're different, we call it heterozygous. And in the case of eye color, the observable characteristic, the phenotype, is either blue or brown. Now, let's say we had a parent that had two big B's and another parent that had two little b's. All of the children would have brown eyes. They would inherit one big B and one little b. They'd all have brown eyes because the big B is the dominant gene in terms of eye color. Now, let's say we had two heterozygous parents, big B, little b, big B, little b. One of the four, about a quarter of the offspring, are going to have blue eyes. So yes, two brown-eyed parents can have a blue-eyed child. If you have two little b's and a big B, little b, about half the offspring will be homozygous for the little gene, the little b, and have blue eyes. Well, that sort of awareness filtered into breeding circles, and one of the people who was very much aware of it was Charles Burnham. He was a University of Minnesota biologist and plant breeder, and he'd retired, but he maintained an interest in the chestnut. He'd always been interested in the chestnut, and he was poking around in the library one day, and he came across a bunch of documents about the American chestnut and what had been attempted. And as he read it, he kind of got infuriated. What the heck were these guys doing? And he called up his friend, Norman Borlaug, who you may remember the name. Norman Borlaug later won a Nobel Prize for the development of a new variety of wheat. He's kind of the father of the Green Revolution in agriculture. So Charles called up Norman and said, Norman, why in the hell didn't you guys backcross that thing? And Norman's response was, I didn't know anything about genetics when I was working for the Forest Service. This is from a wonderful book by Susan Frankel that I recommend you if you want to read more about the history of this whole effort. But Burnham got together with some other people, and they started the American Chestnut Foundation. And what they intended to do was backcross that tree. And what that means is they would cross Asian and American trees and then repeatedly cross successive generations with American trees to increase the American component and retain the Asian genes for blight resistance. They wanted to get the upright growth habit, the rapid growth, the ability to reach the canopy, tolerate coldness of the American tree, and retain those genes that would give it blight resistance. And here's a little pictograph of how that would work. You start with a F1 hybrid that's 50% Chinese and 50% American, and then you successively cross that with Americans, building up the proportion of American genes in the tree. And each time you select for blight resistance, and then eventually you go to an intercrossing stage. And when you intercross, this is the first step here. You take a Asian tree, let's say it has big R for resistance, and the non-resistant American tree will give it a little r and little r. The offspring and first generation are all going to be big R, little r. The difference between this function of blight resistance and eye color is that resistance gene is not dominant. So these trees are all going to be intermediate in resistance. So it's important, because they're all intermediate in resistance, select carefully for those that are the most resistant. And then if you take those and backcross them to an American, you need to select again and pick out the ones that are the most resistant, and then cross them again. That's the intercrossing stage. And the idea here is that some of these will inherit both of those big R genes, and you'll have a strongly resistant tree that still has all the American trees characteristics. And the idea has grown and spread, and the American Chestnut Foundation Pennsylvania and New Jersey chapter has 1,000 members, nearly 100 orchards in Pennsylvania and New Jersey. Nationally, there are 586 plantations. Volunteers log in over 50,000 hours a year, and it's probably the most successful, largest in scope example of citizen science that you will find, because these are almost all volunteers, not completely. And it's been a long process, because unlike trying to create a new breed of dog or chicken or fruit fly, you have to wait a while before the chestnut gets to breeding age, at least 8 years. And then there are other factors involved. But most importantly, as a problem, in fact, is that the genetics are more complicated. It's not one gene that controls blight resistance. And it's becoming pretty clear that it's very rare to find one gene that controls more than 10% of a trait. There are some examples that are different from that, but most traits have many genetic controls. And in the early 80s, it was thought that there were 2 genes involved in blight resistance. By the 1990s, it looked like there were 3. So you'd have a genotype that might look like this. And now it's starting to be clear that there are 9 or more areas of the chestnut genome that are involved in blight resistance. And if all or most of these are important, the chances of the backcrossing, intercrossing approach producing a fully blight-resistant tree are vanishingly small. Here's what typically happens. This is an American tree that's been repeatedly blighted, and it's now turned into a shrub. The central stem has long since died. It keeps sprouting from the roots. They get infected, it sprouts again. This here is an example of a backcross tree, and you can kind of see by looking at the bumps on the trunk that it's fighting the blight, but maybe not too well. It's starting to sprout from the roots. And this is a first-generation Asian American hybrid, much like the trees on Haverford campus. And those trees have some blight resistance, more so than most of the backcross trees. The best of the backcross hybrids are only about as resistant as an F1 Asian American cross. So what else is going on? There's a number of other efforts. And one of them is an effort that I'm involved with, where we're starting with 50% crosses, and then we're going right to the intercrossing stage. One of the things that's interesting about this one is we include a bunch of Japanese trees, which the C. crenata, which most of the early breeding efforts did not. They seem to have a lot of promise. However, if there's more than 3 genes involved, the premise on which this idea was based, probably questionable. However, the third generation hybrids show a lot of segregation of genes, and they may yield valuable data on genetics of other traits. And then there's another organization that is strictly focusing on surviving Americans and crossing them, and here's their website. Here's a picture of one of my plantings in northern Pennsylvania, and you can see that they are a motley crew. These are F2s that have been allowed to intercross from F1s, and they're all different. Look, this tree has burrs on it. A lot of these trees have succumbed early to the blight. Some of them seem to have a lot of blight resistance. Here's one that I'm especially interested in. This is the Japanese American hybrid. It shows very good upright growth habit. Whether it'll maintain that and reach 100 feet tall, who knows? I won't live to see that. I'm trying to eat well and get enough sleep, but it's not. I'm not going to see this tree reach maturity unless it gets blighted first. And that's why, when you're working with trees, the shortness of human life becomes kind of impinges on you a little bit. We're in it for the long haul here. Another effort involves biological control of the blight fungus. There's some promise. This has worked in Europe. Doesn't seem to be working in this country very well, probably because there are too many strains of the blight fungus. But it has a lot of promise. The idea is to make the blight itself sick and spread a disease that would weaken the blight. And there are a lot of people working on it, and it may yet succeed. But I think the big thing that's going on now is genetic engineering, and it's a red flag to some people, and they just don't want to hear about it. But we have to realize that it's been used for over 40 years in human health. And this use has met with little or no outcry, in part because human health has been considered important enough to have the best that science can offer. And it may be time to start considering that we need to do that for forest health. And this is an article that my colleague, Rex Parker, and I wrote, in which we make that case. We basically are saying, look, we're using it in medicine. Let's use it to protect forests because they're important to our long-term health as well. There is a difference in if you're going to do genetic engineering with a tree, it has to go into the germline. The tree has to be able to reproduce and carry that genetic improvement to its offspring. And that's a big difference between what we do when we're creating a medicine that a person consents to take, and that's kind of the end of it. So yes, it's a different situation, but maybe not that different. And there is a transgenic American chestnut in the works right now. It's been found that the blight fungus excretes oxalic acid, and researchers at SUNY Syracuse School of Environmental Science and Forestry, Bill Powell and his team, and Charles Maynard, who's now retired, but they succeeded in taking a gene from the wheat plant, which is a oxidase gene. It enables the tree to produce the enzyme oxidase, and it turns out that the blight fungus excretes oxalic acid, and that kills the tissues of the tree, and then the fungus feeds on those dead tissues. So if the tree can denature oxalic acid, it can resist the fungus. And they've succeeded in getting that gene into a tree. It's called the Darling 58, their latest iteration of that. And here's Dr. Powell in his lab. And this gene, it's present in many other food plants. It's not new material in the diet. And the agrobacterium process appears to have naturally created many transgenic plants. This is an interesting article that makes that case. And it does not seem that the transgenic chestnut is any different in ecological function between wild type trees. Right now, and yes, the trees with the OXO gene are strongly blight tolerant. Here's a little picture here's an American chestnut that's been inoculated with the blight. Here's a Chinese chestnut that's been inoculated with the blight. They definitely are somewhat resistant, but not totally. And here's some of those genetically engineered trees that have been inoculated with the blight. And it seems as if it affords strong blight tolerance. And so right now, it's before the USDA. They're preparing an environmental impact statement. If they approve the tree for release, pollen from these trees could be used to pollinate surviving regionally adapted American trees, and the offspring, about half of them would inherit that gene. And here's an example of how the female flowers on the chestnut can be pollinated. This slide has pollen on it. So if this transgenic pollen gets approved, we could pollinate surviving trees that are fully American and have offspring that are still essentially 100% American, except they have that added gene. There's other genetics related research underway, looking at the DNA of surviving trees, and looking for ways to build resistance to another pathogen that's common in poorly drained, warm areas. And it may be possible to develop a cisgenic American chestnut as opposed to a transgenic. The cisgenic would have genes from the Chinese tree, from the Japanese tree, that confer resistance. Also, it seems as if it's possible to speed up the process of breeding the tree. One way is to use small stem assays, which is to inoculate a seedling with the blight and see how it behaves. Typically, we've had to wait till trees were 8 or 9 years old to infect them with the blight and see how they reacted to that. But it's now possible. There's a method of doing that with smaller trees, and that enables quick determination of whether you've got a blight tolerant tree or not. Another approach is to subject the trees to long day light cycles, and that allows production of pollen in 1 or 2 years instead of 8 or more. But probably the biggest effort that you folks can take part in, that's underway, is finding and propagating surviving Americans. And this is important because these trees, even though they're few and far between, they're out there. They have DNA that has got to be preserved because they represent regional adaptation. And they could become candidates for pollination with this transgenic pollen if it becomes approved. So there are some big trees around, and here's an example of, here's a map of where people have found surviving Americans. Most of these are stump sprouts, and stump sprouts it's cool to find them. It helps you see this is what a chestnut leaf looks like, but they're not what we're looking for. We're looking for trees that are big and possibly producing nuts. And if you do find a tree, here's what to do. Go onto this website, and there's a report a tree form that you can fill out, and this website will show you how to report that tree that you found. What should you look for? Well, you want to look for an American tree, and here's some different. This is in the leaves. This is an American. This is a Chinese. This is a European. This is a Japanese. This is a chinkapin tree. I was part of a little party that roamed through the Allegheny National Forest several years ago, and here are 3 of us. And here's the first little chestnut tree that we found, and of course it was well photographed. But you can see this is a stump sprout, but it's an American chestnut, and it was quite a thrill to find it. We found some more. There are some trees that can fool you. The American beech looks a lot like a chestnut, the leaf does, but when you look at the bark, you see that it's silvery gray, and that's kind of the tip-off. Chestnut oak, chinkapin oak, they look different, look similar from a distance, but if you spot some acorns, you know it's not a chestnut. Here's the American chestnut leaf. It has a little curl at the end of its taper, and kind of shaped more like a canoe than a rowboat. If you see catkins like this, that's a tip-off that you've got a chestnut. But this could well be a Chinese chestnut, especially if it's in an area where somebody might have planted it. There's a lot of Chinese and Japanese trees all over, and there's some European. In the wintertime, the chestnut holds its leaves, so it's a good time to look for chestnuts. Here's a beech tree, which also holds its leaves. Here's a chestnut. The beech has a silvery bark. If you get up close to the tree, you'll see the difference. If you find catkins on the ground, look up. You might have a chestnut tree over your head. If you find some of these, those are old burrs. There's a chestnut nearby if you find one of these. Here's some fresh burrs. The green ones like this tend to have fertile nuts in them. What we're looking for is trees that are producing fertile nuts, and they have to be pollinated. The bottom row here, these are pollinated nuts. The upper ones are not, and oftentimes a chestnut that's all by itself will not be producing pollinated nuts. It needs to be pollinated by another tree. Here's a burr that's opening. They tend to open as they dry out, and it's got one fertile nut in it and a couple non-fertile. And you can see they're little thin things. They won't grow, but this guy will. Here's a bunch of fertile nuts that I was fortunate to find in the Allegheny National Forest last fall. Here's some survivors. This one's in Virginia. This is the Amherst tree. Here's an orchard of Kelly Amherst. The Kelly is a surviving tree that's probably got some European ancestry in it. That's in Harrisburg. And this fellow, the late, great Dave Armstrong and some other people hand pollinated with Amherst pollen the Kelly tree, and they planted the seeds, and Dave established an orchard of Kelly Amherst trees, and they're pretty much pure American. They have some European ancestry, it appears, but they produce a lot of nuts, and Dave used to harvest them and distribute them by the thousands. And this orchard is still going strong, and I was fortunate to go out there the other week and collect some nuts from these trees. Here's a survivor near the Merrill Creek Reservoir in north central New Jersey. You can see it's punched up into the canopy here. We haven't been able to harvest any nuts from it yet, but who knows? I don't know. This tree might be all by itself. I just found one in Mount Holly, and I'm very optimistic. This has a lot of burrs on it. Maybe they have fertile nuts. Here's the trunk of it, and you can see it's probably a stump sprout that has managed to survive long enough to get big. Here's one nearby where Nora and I live in central New Jersey, in the Ted Stiles Preserve. We'll watch this one too. Here's what we're looking for. This is a American chestnut in full bloom in the Allegheny National Forest, and that's the tree that I was able to collect a bunch of nuts from. Unfortunately, this photo is a couple years old, and this tree is starting to get blighted. Some of the top of it is dying. However, its progeny live on. This is a 3-year-old progeny of that tree, and it's growing like gangbusters. It's growing like an American chestnut. It's over 6 feet tall and on the go. So we'll see how it does. We won't see trees like this in the East. These are American chestnuts in Coeur d'Alene, Idaho. They're beyond the range of the blight fungus. The blight fungus has not crossed the Rockies yet. And you can see this is how the tree will grow if it's in a blight-free environment, and maybe how the tree will grow again when we have a truly blight-resistant tree that's out there. What can you do? Well, learn more, join the American Chestnut Foundation, find surviving American trees, and perhaps most importantly, encourage younger generations to get involved, because we are talking about an effort that's going to span decades and probably centuries before the tree is back in the woods in full glory like it once was. A couple of wonderful books, Jane Frankel's book, Susan Frankel, I'm sorry, and called American Chestnut, and then this one is also an excellent book called Mighty Giants. Good reading in both of these, and there's some great sources on the web. American Chestnut Foundation, the New Jersey PA chapter, and the national organization. During COVID, one of the nice things that came out of COVID is that the ACF started a series called the Chestnut Chat Series, and there's one a month, and they are wonderful opportunities to hear from experts, people that are doing research. And the next one is this coming October, 15th on genomic sequencing. So we'll feature Jared Westbrook, who's the chief scientist for the ACF at the Research Center in Virginia. So I recommend checking this out. And then there's some wonderful TED Talks by Bill Powell. And thank you. And it's been a pleasure to talk with you. And please don't hesitate to email me if you have questions.